Welcome to this next video in which we are discussing inflammatory bowel disease. Okay, so in the first video we discussed the basics about inflammatory bowel disease. We discussed that there are two major forms of inflammatory bowel disease, and we discussed what the symptoms of an inflammatory bowel disease flare-up are. In this next video, we're going to go over some background histology or anatomy, if you like. So we are aiming to discuss the pathophysiology that is believed to underlie both of the two major forms of inflammatory bowel disease. And in order to do this, we need to have a good understanding of the structure of the bowel. So I want to go over the structure of the wall of the small intestine and the structure of the wall of the large intestine, because those are the two major areas of the gastrointestinal tract that are going to be affected by Crohn's disease and ulcerative colitis. So, I'm going to begin by going over the structure of the wall of the small intestine, and then we'll adapt it to what the structure of the wall of the large intestine is, because the structure of the wall of the large intestine is thankfully very similar to the structure of the wall of the small intestine. So we'll come over to this side here. So how am I going to do this? Well, I'm going to draw you the different layers of the wall of the small intestine, and we're going to start from the luminal side. So we're going to imagine that we're in the lumen of the small intestine, and then we are drilling down, and we're going to see the different layers, and we'll work our way right to the outer side of the wall of the small intestine, and then we'll adapt the picture for the large intestine. So, from the luminal side then, so we're imagining we're in the lumen of the small intestine. What is the most luminal layer of the wall of the small intestine? If you're a little man in the small intestine, what's the layer that's going to be facing you? Well, this is going to be the epithelial layer. Now, the epithelial layer that lines the small intestine is a simple epithelium. What does that mean? That means that it's made up of a single layer of cells rather than a stratified epithelium, which is where the epithelium has multiple different layers of cells. And of course, the major example of a stratified epithelium would be the epidermis of the skin. So, we're going to have a single layer of cells for the epithelium of the wall of the small intestine, and these cells are going to be cuboidal, so they're going to be a sort of rectangle shape, but they're not going to be an extreme rectangle shape. Remember, there are two types of rectangular epithelial cells. There are cuboidal epithelial cells and columnar epithelial cells. Now, columnar epithelial cells are a more tall cell. They're 4 times as tall as they are thick, is the general ratio that we give. Whereas cuboidal cells are nowhere near as tall as that. They're much less tall, so they're much less extreme rectangle. So, let me now draw the epithelium. Now, the epithelium is complicated of the small intestine because it's not just a straight line. Of course, what I'd love to draw here, what would be really simple is if I just drew a nice layer now, a nice simple epithelium sat, of course, on a basement membrane, as indeed all epithelia are, of cuboidal epithelial cells, and that was that. However, that's not what I'm about to draw because the epithelium isn't just flat, and you probably are aware of this. Instead, coming off from the surface of the epithelium, you are going to have evaginations, projections upwards, that are called villi. So the entire length of the small intestine, you have these villous projections of the epithelium, which are in order to increase the surface area of the epithelium, because the epithelium is where the absorption occurs. Not only that, not only do you have these complicated evaginations called villi, you're also going to have invaginations of the epithelium that are called the crypts of Leberkuhn, and these are going to house things called the intestinal stem cells that are going to make the epithelium of the small intestine. Because the epithelial cells are continuously turning over. You don't keep the same epithelial cells for that long. They continuously shed off and have to be replaced. So, enough talking, let's actually draw a little bit of this. So I'm going to draw these evaginations and these invaginations. So here is an evagination of the epithelium outwards, like so. That's, of course, a villus. And then in between the villi, you're then going to have these invaginations inwards, which are the crypts of Lieberkühn, and then it could go on. So here's, if you like, the flat level of the epithelium. Here's an evagination outwards, and here's an invagination inwards. And I could go on. I could draw another evagination coming out here, and then another invagination, etc. I'm not going to, because it'll just take up time, and I've shown the two essential components here, the evaginations and the invaginations. So now the time-consuming bit, just bear with me. I have to actually turn this line into cells by making them a thickness here. So these are my cuboidal epithelial cells here, like so. And of course, they're all going to be sat on a basement membrane. All epithelia in the body have a basement membrane, and this is a meshwork of proteins and also carbohydrate fibers. It's a rigid meshwork which all the epithelial cells will be attached to, because you have to question, how do these epithelial cells not just fall into the lumen of the intestine? Well, they do eventually, because they detach from the basement membrane, but the way that they hold tight is by binding to a rigid structure here, and this rigid structure is the basement membrane that I've put in red here. So in red, and I need to go back to a thinner pen. In red, this is the basement membrane, which for short we often just abbreviate down to the BM, but which I have written out its name in full here. Okay, and now I have to somehow find that orange colour back again. I apologise if this is not the exact same shade of orange. You know it's not, but never mind. So what I'm doing now is, of course, separating this layer into the different little epithelial cells here. And these are, remember, cuboidal epithelial cells. So you can see they're just about a rectangle. Their width is just about less than their height. And of course, they would have nuclei. I'll give a few of them nuclei here. Okay, and I could continue this picture on. I won't waste your time by separating all of this into cuboidal epithelial cells and giving them all nuclei, but you get the point. So, to give this epithelium its name then, it is a simple, and I'll remind you that that means that it's made of a single layer of epithelial cells rather than a stratified epithelium where you have multiple different layers of cells piled on top of one another like you would have in the epidermis. And then we describe the shape of the cells. They are cuboidal, and then it's an epithelium, so we call it an epithelium. So simple cuboidal epithelium. Okay, sat upon a basement membrane. Now, before we move off the epithelium, before I do what I just wanted to do, let me label up these projections outwards and these projections inwards. So this is called a villus here, and of course you'll have loads of villi. This is the projection outwards. So this, if you like, is the flat sort of standard surface of the intestinal epithelium, and then we have this projection outwards, which is a villus, into the lumen, which of course is this space here, and then we have a projection into the deeper layers of the wall of the intestine, which is called a crypt of Lieberkühn. And I want to explain to you where the intestinal stem cells are in just a moment. So this is a crypt of Lieberkühn. Okay, so more information about this simple cuboidal epithelium then. So there are actually two major types of cells within this simple cuboidal epithelium. There are actually far more than that, but to keep things simple, there are two major types of cells within the simple cuboidal epithelium. And these are the actual enterocytes, which are the major ones. These are the ones whose job it is to absorb nutrients from the intestinal lumen and pass it over into this underside here where there will be blood vessels, and we'll go into that in more detail in just a moment. So most of these cells are going to be enterocytes. So let's say all of these ones here are going to be enterocytes, and these have little projections out from their surface known as microvilli, which I'm trying to draw on here. So if I was to take in a zoomed picture of an enterocyte, it would look something like this. So here is its cuboidal shape, and then from its luminal surface that faces into the lumen of the intestine, it has these projections into the lumen, and again these are to increase the surface area for absorption. and these are known as microvilli. So do not confuse the villi, which are actual evaginations of the entire epithelium of the wall of the intestine, for the microvilli, which are just these projections outwards of the actual enterocytes. So, most of the simple cuboidal epithelial cells will be enterocytes. Their job is to absorb nutrients, and they will have these microvilli. However, the second type of cell that's within the simple cuboidal epithelium that I'm going to tell you about, as I say, there are more, but we're not going to go into them, is what's called a goblet cell. Now, the goblet cells have a slightly different function. Their function is not to do with absorption of nutrients across the intestinal epithelium. Instead, their function is in secreting mucus. So let's turn this one here into a goblet cell, and these are a rarer cell than the enterocytes, and they're scattered around the enterocytes. So I might have one here as well. I'm just colouring them in green to show them. They would have nuclei as well, but I won't put those on. And they're still cuboidal in nature, but they don't have the microvilli anymore. And their job is to secrete components of mucus. In particular, they secrete things known as mucins. So they have vesicles within their cytoplasm that are full of these things called mucins, and these are a component of mucus. And you can either spell mucus with an O there, or you can drop the O and just have US. Now, where is the mucus then? So what I haven't told you that is that the entire epithelium is covered with a layer of mucus, which I'm putting in here in green. So this is representing a layer of mucus. Now, this is to lubricate the epithelium so that everything can move through the intestine more nicely. Now, when you're making mucus, it's a complicated thing to make mucus. You firstly have to put some fluid on your surface, and that fluid will have salt, it will have electrolytes in, and that's called a serous secretion. So, in order to make mucus, you firstly need to make a serous secretion, which is water with electrolytes in. It's the same sort of stuff that extracellular fluid is. Now, the enterocytes will secrete a serous secretion onto their apical surface, onto their luminal surface. So these enterocytes, they put onto their surface a serous secretion. Now, that is not yet mucus. To turn that into mucus, you have to add in special things, such as mucins, that will make it much more viscous and will turn it into mucus. So I can actually just draw a little explanatory flow diagram here. Serous secretion can become a mucus secretion by secreting mucins into it, and it is the goblet cells that have this function of secreting these mucins. So they make the mucus effectively. They turn the serous secretion that the enterocytes produce into a mucus secretion by adding in these mucins. So goblet cells are very important for making this fluid that is on the luminal surface of the epithelium a mucus secretion rather than just a serous secretion, and that of course is better for lubricating the epithelium than just a serous secretion. It's also quite good at protecting the epithelium from all the bugs that are in the luminal contents. Many of which are commensals, and which are going to actually be extremely important, or are believed to be extremely important for the pathophysiology of inflammatory bowel disease, but we'll come back to those later on. Okay, so we have seen these two major cells that are part of the simple cuboidal epithelium then, the enterocytes and the goblet cells. Let me now talk about the intestinal stem cells, because this is a interesting little story to add in here. So, these epithelial cells, these simple cuboidal epithelial cells, both the enterocytes and the goblet cells, they do not have a very long life, I'm afraid. They get to live for a few days and then they are shed away. This is because this epithelium is very prone to getting cells removed from it. Remember, stuff is moving through the lumen of the intestine. It's continuously rubbing against the epithelium. So this epithelium has to be continuously replaced. It's like the skin is continuously replaced. You cannot just make one skin epidermis and expect it to last you your entire life, because, of course, you're exposing it to the harsh environment, and cells will be lost. So you do have to continuously replace it. The same goes for the epithelium that lines the gastrointestinal tract, and indeed the small intestine is part of the gastrointestinal tract. So, right at the bottom then of the crypts of Lieberkuhn, which is a safe place for them to be present, are some cells that aren't replaced. These have to be kept safe, therefore, which is why they're right at the bottom of the crypts of Lieberkuhn, of which we have an example here. So these are really important cells that, as I say, can't be replaced and which are responsible for continually producing more epithelial cells. So these are intestinal stem cells, and of course, since we're in the small intestine, you could call them small intestinal stem cells. So these are right at the bottom of the crypts of Lieberkuhn. They're cuboidal in shape again, and their job is to divide and divide and divide. So let's have an intestinal stem cell here. So what it does, all it does is divide and produce two cells. So it continuously divides to produce two cells like so. And what happens is that one of its offspring cells remains a stem cell. So let's say this one is going to remain a stem cell. And the other then undergoes a differentiation process. So this one now undergoes a differentiation process, which means that it's going to change the type of cell it is, and it can become one of these cells of the simple cuboidal epithelium above. It can either become an enterocyte or a goblet cell, or the other types of cells that are more specialist knowledge that I haven't gone into because it's not necessary for our story, for our study of inflammatory bowel disease. Okay, so these intestinal stem cells, they continually divide, they produce a new cell that stays in their position and keeps the population of intestinal stem cells the same, and then they produce another one that comes off upwards, and which will begin the differentiation process and might become an enterocyte or a goblet cell. So some of the cells that are being chucked off by the intestinal stem cells will become enterocytes, some will become goblet cells, and these gradually make their way up the crypt of Lieberkuhn epithelium here, and up the villus, and then, of course, they will, by this point, be the cell that they've differentiated in. As they go up the crypt of Lieberkuhn here, they'll be gradually differentiating, and then once they get up here, they will have finished the differentiation process, and they'll either be an enterocyte and a goblet cell, and there is a continuous sort of moving along of the epithelial cells. They continually move upwards, and then the ones at the tips of the villi are shed away. So this is the cycle of the intestinal epithelium then. At the tips of the villi, cells are being shed away. That means that these ones have to move up to replace them, and therefore everything has to move up somewhat. At the bottom, we would then have a gap, except for the fact that the intestinal stem cells are continually dividing to produce new cells to replace the ones that are being lost. So there is a continual motion up from the crypts of Lieberkuhn and then up the villi to be shed off. And that is, I'm afraid, the short story of the epithelial cells of the small intestine. Okay, so I think that now covers the story, all that I want to tell you about the actual epithelium that lines the small intestine. Remember, it's a complicated structure with these villi and these crypts of Lieberkuhn. And this goes for all the different portions of the small intestine, the duodenum, the jejunum, and the ileum. Let's now continue the story onwards. So we're going to now go underneath the basement membrane. And remember, the basement membrane is a meshwork, a spider's web of fibres, protein fibres and carbohydrate fibres that forms a rigid structure for the epithelial cells to latch onto. Underneath the basement membrane, we then have a layer of connective tissue that is known as the Laminar Propria connective tissue. So let me just decide on an appropriate colour to have this in. So I'll demarcate the lower boundary of it here. So all of this space here, including all of the space that's in the villus here, all of this space here, this is going to be this Laminar Propria layer. And I'm just going to come a little bit this way so I can add the caption on here. So this is the Laminar Propria, and this is just connective tissue. So you're going to have loads of collagen fibers, loads of protein fibers, loads of carbohydrate fibers, all of the fibers that make up extracellular matrix. You're going to have loads of those in this position. And running through that connective tissue, you're going to have very important structures, which are the blood vessels and the lymphatic vessels. And this is really important because remember, what is the function of the intestines? What is the function of the gastrointestinal tract? It is to absorb nutrients. The enterocytes are involved in passing nutrients from the intestinal lumen to their basolateral side, and those nutrients need to go into blood vessels or lymphatic vessels, so that they can go into the circulation and feed the rest of the body. So in this laminar propria, you're going to have arterioles, venules, and capillaries in between them, and you're also going to have lymphatic vessels called lacteals. So let me get some good colours to do this in and show you this. So I'm going to actually get a thicker pen to show this. Actually, not that thick. So here, this is representing an arteriole, hence why I have chosen a bright red colour to do it. And you can see that it goes right into the villus there. Now let me get a venule, and of course, keeping classical, here is a venule in blue here. And then in between those, which I won't draw, would be loads of capillaries. But you can imagine, in between them, you'd have loads of little capillaries connecting the arteriole, which is a tiny little arterial blood vessel, to the venule, which is a tiny little venous vessel. So let me just label these up. So this is a tiny little arteriole, and we also have a tiny little venule there. And in between them, we'll have capillaries. In fact, I will try and show a few capillaries. Why not? So in yellow here are a few little capillaries in between them, which are the connecting blood vessels, even smaller. And of course, the capillaries are going to be the major site where nutrients are going to come in. So nutrients from the enterocytes will go into the capillaries, and then of course the blood is moving into the venule, and then we'll see where they're going later on. Now, that's not, however, the only blood vessels that you're going to have in... Sorry, they are the blood vessels that you're going to have in the villus, but you're going to have another type of vessel, which is a lymphatic vessel. And I'm going to draw this in this sort of mint green colour. Lymphatics are often showed in green. So also in a villus, you'll also have a lacteal, and I apologise that this seems to be going more luminal than the basement membrane. I didn't mean for that to happen. I just didn't want to have to put it over the arteriole and the venule here. But imagine that that's also underneath the basement membrane. I appreciate that it's actually going over the basement membrane in my picture. That is an unfortunate mistake, and I can't really do anything to rectify. So this is what's known as a lacteal, and this is a lymphatic vessel. So this is going to drain into lymphatics. It's going to go into bigger and bigger lymphatic vessels through the lymph nodes, and eventually the lymphatic system empties into major veins up in the top of the chest. Now, nutrients are going to not only come into the capillaries, but also the lacteals. In particular, lipids are going to come into the lacteals. So these are often described as carrying a substance that resembles milk, because lots of the lipid structures that come in through the enterocytes end up going into the lacteals rather than going into the capillaries. So sugars and amino acids are going to go into the capillaries, but lots of the lipids go into the lacteals to drain into lymphatics rather than draining into the portal venous system, as indeed these venules will feed into. Okay, so that's what the laminar propria is. It contains lots of connective tissue, and just to make this a little bit more obvious, I will colour this in here. So all of this is not just empty space, it's going to be connective tissue with the blood vessels moving in it, like so. Okay, then another little layer to talk about here. Underneath the laminar propria, you have a very thin layer here of smooth muscle cells, which is known as the Muscularis Mucosa. So this is a very thin layer of muscle. You're going to have a much thicker layer of muscle later on, but this is just a little curious thin layer of muscle here known as the Muscularis Mucosa, and this will be smooth muscle cells, and I'll try and draw a few of these smooth muscle cells in here. So I'm drawing them in their classical spindle shape with the thick bellies and the thin edges. So these are smooth muscle cells, and it's really just a single layer thick of smooth muscle cells, so it's not a thick layer of muscle at all. Now, muscularis obviously just refers to muscle. Mucosa, what does this refer to? This refers to the fact that it's the muscle layer of the mucosa of the small intestinal wall. So what does the mucosa mean? All of the layers that we've discussed now, collectively, they are called the mucosa of the small intestinal wall. So the epithelium, the basement membrane, the laminar propria, and the muscularis mucosa, collectively you can refer to all those layers as the mucosa of the small intestinal wall, or as, when we adapt this to the large intestine, the mucosa of the large intestinal wall. Okay, so that's the muscularis mucosa. What is underneath the muscularis mucosa? You have another layer of connective tissue that is known as the submucosal connective tissue, or the submucosa. And now you see why I wanted to tell you that piece of terminology about the mucosa, because now you can understand why this is called the submucosal layer, because it's underneath the mucosa. So here we have lots of connective tissue. I won't colour it in full yet because I want to put the blood vessels in first. So what does this contain? It contains loads of really important blood vessels, bigger blood vessels this time. So it contains the bigger blood vessels that these smaller arterioles are coming off here. So I'll put one of these in here. So here is a big arteriole. It might even be so big that you might describe it as an artery now, but it's bigger than this arteriole here. Depending on its size, you would either call it an arteriole or an artery. And then, of course, you'll have bigger venules bordering on actually being called veins now, and I'll try and connect my venule onto that one. Okay, so the branches that are going into the laminar propria, penetrating through the muscularis mucosa to get into the laminar propria, come off bigger blood vessels that are in the submucosa, and of course you're going to have bigger lymphatic vessels as well within the submucosa. So let me just draw these on. So here is now bigger lymphatic vessels, or a bigger lymphatic vessel rather, which my little lacteal from the centre of the villus is now going to drain into. So the large blood vessels and the larger lymphatic vessels that supply these smaller branches that penetrate through the muscularis mucosa and go into the laminar propria are within the submucosa. So that's what the submucosa contains. And again, it's just a layer of connective tissue here. So loads and loads of protein fibres and carbohydrate fibres that are forming a great big web all over the place. And of course, it will also contain the cells that actually produce all that connective tissue, which are the fibroblasts. I might just put a fibroblast in the laminar propria. So fibroblasts have a sort of spindle-like shape as well here, and I'll put its nucleus here. So that is representing a fibroblast. And these are the cells that can produce the components of the extracellular matrix. So they produce the collagen fibres, the carbohydrate fibres, they produce connective tissue. So dotted around the place in the laminar propria and the submucosal layer, you'll have fibroblasts. Okay, now we're going further out. We're penetrating further and further out of the wall of the small intestine here. This is where we get to the proper muscle layer, the thick muscle layer that's actually involved in peristalsis, i.e. which contracts to move the contents of the lumen forwards. The muscularis mucosa is a tiny little layer that doesn't really do that much. This is the proper layer of muscle. So I'll give you a nice thick layer here. And this layer is called the muscularis, which of course we know means muscle. And then to show you the fact that this is the proper layer, we call it the Muscularis Propria. Okay, now the Muscularis Propria, again, it is smooth muscle cells, so it's not skeletal muscle cells, it's always smooth muscle cells in the bowel, and we split it into two different layers, and these two different layers have the smooth muscle cells oriented in different directions. So, the innermost layer is the circular layer, and these smooth muscle cells are actually going to be oriented to form rings that go around the circumference of the small intestine tube. So if I just draw a little picture over here, so this is representing a portion of small intestine, a very simple picture, it's just a tube. This there is going to have smooth muscle cells that are oriented in rings going around there. So if we imagine that this section that I have drawn here was actually a section going along like this, so I took, a little section of the wall like so, i.e. I took a section and I'm looking at it, I've cut through so that it's going in the longitudinal direction, then the way I would see these smooth muscle cells is I'd have cut through their bellies maybe. So I'd just see them like this. I wouldn't see their full lengths because they are oriented perpendicular to the direction I've made the cut. Okay, so this is the circular layer. The smooth muscle cells are oriented in rings, like so. They form rings going around the circumference of the small intestinal tube. Whereas this outer layer is then the longitudinal layer, and in this layer the smooth muscle cells are actually oriented in the longitudinal direction of the small intestine. So again, if I had taken a section in the longitudinal section, then I would now see the smooth muscle cells in their full length, basically, because I've cut in the same direction as they are actually oriented. So here they are, and of course they would have nuclei as well. So I'll give them nuclei, and I missed the nuclei out up when I drew the Muscularis Mucosa up here. Okay, so you have these two different layers of the Muscularis Propria, the circular ones and the longitudinal ones. And of course, when the circular ones contract, they are oriented to form rings around the circumference. That means these rings will constrict down, and therefore the entire lumen of the small intestine will constrict down. And you can imagine that that can push food forward. When the longitudinal ones constrict, you can imagine that what that will do is it will mean that the intestine contracts down in this sort of direction. This bit will be pulled this way, this bit will be pulled this way, and again, you can imagine how that might be useful for moving food forward. So both of these are very important for moving food forward, for peristalsis as it's called. So peristalsis is the name we give for this moving of the contents of the intestinal lumen forward through the contraction of the Muscularis Propria layer. Okay, so we're nearly done now. One final layer of the small intestinal wall. If we come right to the outer side, what do we hit? We hit the layer of the visceral peritoneum, which is known as the serosa. So this is the layer of the visceral peritoneum. So remember, all of the intestine is covered by a layer of the peritoneal cavity, which is the visceral peritoneum. And this is a layer of very, very flat cells that are similar to epithelial cells, but which are not called epithelial cells because of their embryological origin. Instead, they are called mesothelial cells. So you can also call this a mesothelium. So you'll have a basement membrane. They're sat on a basement membrane, as indeed all epithelia and all epithelia-like structures are. So we'll have a basement membrane here, and then sat on top of that basement membrane, you then have the actual cells themselves, and they're little tiny squamous cells, like so. So they're very flat. So these are squamous cells. So all around the small intestinal wall, you'll then have this serosa layer of visceral peritoneal cells or mesothelial cells, like so. Okay, so these are the serosa cells, and then after that you'd be in the peritoneal cavity, so there would be fluid here, peritoneal fluid. Right, so there we go. There is the wall of the small intestine. Those are the different layers of the wall of the small intestine. And the really important bit to understand is the lamina propria, because that's the major place where inflammation is going to occur in inflammatory bowel disease. In Crohn's disease, it can go a little bit deeper. It can go into other layers, such as the submucosa, and even sometimes into the muscularis propria layer. But for simplicity, the major place that the inflammation is going to occur is within the lamina propria, and we can see that all of the blood vessels that we need to bring troops out of the bloodstream, so the inflammatory exudate and the white blood cells, are present here. Okay, so finally, to complete up this video, let's just talk about how we could adapt this picture to make it relevant for the large intestine rather than just the small intestine. So, we drew the large intestine. We drew the small intestine. The large intestine is very similar, except that you would not have villi anymore. The large intestine does not have villi the way the small intestine does. So just get rid of the villi. Imagine that this is now flat, still with the crypts of Lieberkühn, but no longer with the villi, and that then is the picture that we would have of the large intestine. Everything else is exactly the same. A little bit of terminology changes. You no longer refer to the major cells of the epithelium of the large intestine as enterocytes. Instead, you call them colonic epithelial cells. So we refer to them as colonic epithelial cells, and I'll just have ECs for epithelial cells. But they still have microvilli. So this is why, once we've studied the small intestine, it's very easy to then understand the large intestine, because really it's just an adaptation of the small intestine. Take away the villi and just have a flat epithelium here with still the crypts of Lieberkühn with the intestinal stem cells creating the epithelium, and then you've got the large intestine. So we'll have a break here, and in the next video we'll study the pathophysiology of inflammatory bowel disease.